Florida dumped 500,000 tons of oyster shells offshore. And what formed next left scientists scrambling for answers. These weren't shells guided by precision or a sophisticated plan. They were leftovers discarded, scraped off plates and shoveled out of seafood plants, loaded onto barges, and tipped into the Gulf of Mexico between 2007 and 2024. Nobody expected anything remarkable. Critics called it reckless ocean dumping. Fishermen feared it would destroy what little remained of Florida's struggling coastal ecosystems. Even the scientists directly behind the program braced for failure. But something happened on that Gulf seafloor that violated every expectation, every timeline, and every assumption about how nature works. A dead stretch of ocean bottom began transforming into something alive, faster, denser, and more complex than anything the data predicted. What formed down there wasn't just a reef. It was something still expanding today. And exactly how it happened will change the way you think about the ocean. The ocean experiment nobody believed would work. Between 2007 and 2024, the Florida Fish and Wildlife Conservation Commission ran one of the most controversial coastal experiments in state history. The premise was almost absurdly simple. Collect discarded oyster shells from restaurants and seafood processing plants across Florida. Truck them to the coast, load them onto barges, and dump them into the Gulf of Mexico near Cedar Key. Over 500,000 tons of them. We're dying. The water quality in general is dying from 1,000 little cuts, and then we have one big gash in the middle with the discharges going on. The opposition was immediate and fierce. Environmental groups called it ocean dumping disguised as conservation. They warned the shells would smother whatever fragile life remained on the soft, sandy bottom. Local fishermen who had spent generations working those waters feared sediment burial, disrupted currents, and permanent destruction of fishing grounds their families had relied on for a century. The criticism wasn't fringe. It was loud, credible, and backed by real ecological concern. And yet, the commission pressed on. Their logic was stark. These shells were going to landfills anyway. If there was any chance they could function as reef substrate, the experiment was worth running. Here's the catch. Nobody, including the scientists who designed the program, actually expected it to work this fast. When the first monitoring teams returned to the deposition sites 18 months after the initial shell drops, they were braced for the worst. What they found instead made Dr. KSKY, a shellfish habitat specialist with the Florida Fish and Wildlife Conservation Commission, stop mid-sentence during a team debrief. Life was colonizing the shell mounds. Not in 2 or 3 years, the standard timeline for any artificial reef project. In months, clusters of marine organisms were forming at rates that had no precedent in the scientific literature. The shells weren't acting as passive debris. They were functioning as biological triggers, and nobody could fully explain the speed of what was unfolding. But here's what nobody expected. To understand why it was happening so fast, you had to look at something so small it was effectively invisible. And what was happening down there was stranger than anyone had imagined. The invisible phase, microlife that rebuilt an ecosystem. The pioneers weren't fish. They weren't oysters. They were bacteria. Within days of the shells hitting the seafloor, microscopic bacteria began colonizing the calcium carbonate surfaces, forming thin biofilms, structured living carpets that transformed dead shell into functional microhabitat. These weren't random bacterial patches. They were organized biological infrastructure, laying the groundwork for everything that would follow. And get this, the shells were doing their own chemistry. Oyster shells are composed primarily of aragonite, a form of calcium carbonate that dissolves slowly in seawater. As those shells released calcium ions, they created tiny alkaline microzones in the surrounding water. Those localized chemical shifts were exactly what calcifying organisms, barnacles, tubeworms, and juvenile oysters needed to attach and grow. The shells weren't just providing physical surface. They were manufacturing the conditions for their own colonization. Weeks later, diatoms and microalgae moved into the biofilms, creating the base of a rudimentary food web. Microscopic crustaceans and larval mollusks followed. Those grazers drew the first juvenile fish. Each stage fed the next, a self-reinforcing loop building itself invisibly out of discarded seafood scraps. Here's what that means. Sandy seabeds cannot do this. Sand offers no attachment surface, no chemical microzones, no structural complexity. The soft bottom Gulf floor was biologically trapped. The shells bypassed that trap entirely. By the end of the first 6 months, shell surfaces were coated in layered biofilm and microalgae. To a casual observer, the sites still looked like failure, but Dr. Krimsky's water samples and sediment cores told a different story. In less time than a traditional artificial reef shows any biological response at all, the shell deposits had established a complete microscopic ecosystem. The invisible phase was the hidden scaffolding. Without the bacteria, without the aragonite chemistry, without the biofilm feeding the first grazers, there is no reef, just calcium, just sand. The scaffolding was in place. If this is already blowing your mind, hit subscribe because what came next made the scientists question everything they thought they knew about how reefs are built. The return of the reef builders. Oysters need hard surfaces to survive. That is the fundamental constraint that had been quietly strangling Florida's Gulf Coast for decades. Oyster larvae, called spat, drift in the current searching for substrate. In a healthy reef system, that substrate is the accumulated shells of previous generations. Centuries of dead oyster layered into a hard, complex platform, filtering water, preventing erosion, and providing a home for an estimated 300 different species. But Florida's natural reefs had been gutted. Over-harvesting, pollution, and coastal development had dismantled a network that once spanned hundreds of square miles. By the early 2000s, larvae were drifting through open water with almost nowhere to land. No substrate, no settlement, no new oysters. When the fish arrived, everything changed. Reef fish don't wait for an invitation. The moment structural complexity appears on a featureless seafloor, they find it. Within 2 years of the reefs establishing themselves, fish surveys near Cedar Key were returning numbers that made Dr. Pine's team recheck their methodology. Biomass around the reef sites had increased by more than 340% compared to control areas. Species diversity was up 280%. These weren't fish aggregating from somewhere else. They were juveniles surviving in habitat that hadn't previously supported them. The reefs were producing life, not concentrating it, producing it. Redfish arrived first, tucking into the complex shell structure to feed and hide from predators. Sheepshead followed, thriving on the invertebrate communities, coating the hard surfaces. Flounder worked the edges. Snook appeared in numbers not seen in those waters in years. And get this, Mike Lenz wasn't the only captain who reversed his opposition. Charter operators across Cedar Key began formally requesting new reef sites near popular fishing grounds from the same commission they had fought against 6 years earlier. Hotels, bait shops, and waterfront restaurants all reported measurable revenue increases tied directly to the marine surge around the reef sites. The ecological cascade continued upward. Octopuses established territories in the reef's complex gaps. Sea turtles reappeared, foraging on the invertebrate communities. Dolphins were observed hunting systematically at the reef edges, working the dense fish concentrations. Every new arrival reinforced the food web beneath it. The system was growing more complex, more stable, and more resilient with each passing season. What made this ecologically significant, not just impressive, was what the data confirmed about the nature of the change. Dr. Pine's team was careful to distinguish between fish aggregation and fish production. Artificial reefs, made of concrete or sunken ships, are notorious for concentrating fish that already exist in an area, essentially redistributing a fixed population around a new landmark. That can look dramatic in a survey without representing any actual increase in marine life. The Cedar Key reefs were different. Juvenile fish counts, animals born in those waters and surviving to maturity there, were the primary driver of the increase. The reefs were producing fish, creating the conditions for survival that hadn't existed before. That distinction mattered enormously for how Florida's fisheries managers thought about the future of the program. But fish were only part of the story. Something else was happening to the water itself. something nobody had predicted, and that would have consequences reaching far beyond the reef. The billion-gallon cleaning machines beneath the waves. One adult oyster filters between 30 and 50 gallons of seawater every single day. Run that number across hundreds of thousands of oysters thriving on the restored reefs. Entire reef systems were now processing billions of gallons of Gulf water daily, extracting suspended particles, excess nutrients, harmful bacteria, and agricultural runoff with no machinery, no electricity, and no maintenance cost. Nature had built an industrial-scale water treatment plant out of discarded restaurant waste. The Gulf Coast has a chronic runoff problem. Nitrogen and phosphorus from farms and urban development pour into coastal waters constantly, fueling the harmful algal blooms that cloud the water, suffocate marine life, and periodically trigger mass fish kills. For decades, those blooms had been a defining feature of degraded Gulf ecosystems. The oyster reefs were attacking that problem at the source. Here's what that means. Water clarity around the reef sites began improving measurably within just a few years. Oyster shells crammed into baskets offer a new defense against waves, tides, and storms. Dissolved oxygen levels rose. Nutrient concentrations dropped, and the improvements weren't staying local. They were radiating outward, affecting surrounding waters in a growing radius around each site. Clearer water lets sunlight penetrate deeper. Seagrass, critical habitat for juvenile fish, manatees, and sea turtles, requires light to survive. As the reefs cleared the water, seagrass beds began expanding into zones that had been too murky for them for years. And get this, seagrass then stabilized sediment, which reduced turbidity further, which let more light through, which enabled more seagrass growth, a second self-reinforcing feedback loop triggered by the first, radiating benefits across the entire coastal system. Researchers estimated the filtration services alone generated by the 500,000 tons of shells deposited between 2007 and 2024 were worth tens of millions of dollars, value that would have required extraordinary mechanical infrastructure to replicate. Nature was doing it for free. The results weren't staying in Florida. Alabama launched its own shell recycling program by 2015. Mississippi followed. Louisiana initiated massive reef-building initiatives. The Billion Oyster Project in New York Harbor adopted the same principle in one of the most polluted urban waterways in North America. The model was spreading because it worked. The concept emerging from all of this had a name that researchers began using with increasing frequency: circular environmental design. The idea that materials once considered waste, oyster shells, coral fragments, limestone rubble, could be repurposed as functional ecological infrastructure, not as a stopgap, but as a genuine design strategy. Florida's project hadn't just restored a reef. It had demonstrated a philosophy that was beginning to reshape how conservation engineering approaches the problem of degraded coastlines. International interest followed quickly. Coastal engineers in Canada, Australia, and Europe began studying the Florida model, seeking nature-based solutions to problems they had always addressed with concrete and steel. Traditional artificial reef programs, sunken ships, and reef balls had a consistent failure mode. They concentrated fish that already existed nearby without generating new biological production. The underlying cycles of reproduction, filtration, and habitat creation were absent. Themselves around the Gulf, the reefs weren't aggregating existing marine life. They were generating new life continuously, season after season, without any help. The long-term trajectory pointed towards something restorationists rarely claim: a self-sustaining system that no longer requires human management. The shell deposits of 2007 had seeded a cycle, now perpetuating itself indefinitely. Each oyster that lived and died contributed substrate for the next generation. Each generation expanded the reef's footprint. Each expansion created new habitat, filtered more water, protected more shoreline, and supported more species. The intervention had ended. The ecosystem had not. It was running on its own, biological momentum, and the data suggested it would continue for decades, potentially centuries to come. That outcome had been unimaginable in 2007. It was measurable fact by the early 2020s. Should every coastline in the world be doing this? Or are we missing something the Gulf is about to tell us? Hit subscribe and check out the next video on your screen, because the science on where oyster restoration goes next is even more extraordinary than what you've just seen. 500,000 tons of oyster shells discarded, written off, tipped into the Gulf without any certainty that a single useful thing would result. And look what formed.